All right, ladies and gentlemen, welcome back. We are going to continue our discussion today on macromolecules by talking about proteins. And proteins are probably one of the biggest workhorse molecules in your body because they do so much. So let's talk first and foremost just about their sort of monomer-polymer structure. So when we look at proteins, they are polymers, excuse me, that are built of monomers called amino acids. And amino acids are what you're looking at here in the picture. Amino acids, when we look at them structurally, really are kind of four major component parts centered around an asymmetrical carbon in the center. So when we look at your asymmetrical carbon here, remember carbon can make four bonds. So one of those bonds off that carbon will always be a hydrogen. The other will be an amino group, so an NH2. The other will be a carboxyl group, which is a C double bonded to an O and an OH. And then the fourth bond will go to an R group. And this is actually where that term amino acid comes from. So here's your amino group, and carboxyl groups tend to make the molecules acidic. So there's where the amino acid term comes from. That R group is going to be really important because that is what is different. So when we look at amino acids, there are 20 different types. And when we look at the bottom part of this molecule, this is identical to every amino acid, but there are 20 different side chains or R groups. And that R group is really what's going to determine the properties of that amino acid and how it plays its role in the overall protein structure. So when we look at those amino acid side chains, they get labeled into or lumped into a set of categories based on the way that they function. So this particular set of amino acids, their R groups are all nonpolar, so they're hydrophobic. So when we look at the way that they build into the protein structure, these guys are going to want to bury themselves in the middle of the three-dimensional protein structure because they don't like interacting with water. And remember that your cellular environment, both internal to the cell itself and then also externally, is just surrounded by water. And so more often than not, these particular amino acids end up buried in the internal structure of a protein as a way to avoid that watery environment, just like a phospholipid fatty acid tail buries itself in the middle of your phospholipid membrane to avoid the water for the same reason. This group, these guys are polar side chains. So if you take a look, a lot of them have an OH group somewhere that will often lose that H, and that oxygen will end up being ionized as a result, or they're capable of doing some other things as well. But these guys are polar, so these are hydrophilic, in which case they like interacting with the water. They'll do that hydrogen bonding or something along those lines. And so we'll often find these guys on the exterior structure of a three-dimensional protein structure. And then we can also have acidic and basic ones. So if you look at the aspartic acid and the glutamic acid, these guys are negatively charged because they'll lose those hydrogens. And then you have your basic or positive ones, and if you look, they all have nitrogen groups hanging off here that end up either taking on an extra hydrogen or losing that hydrogen somewhere along the way. They can gain those extra positive charges there. So these guys are also hydrophilic and will often also be on the outer exterior structural component to a protein. Now, do you need to memorize all of these? No, but if you can start recognizing pieces of them and what sort of role that they'll play, it does become helpful the more that you study this over long term. So these guys will then end up linking together. So when we talk about individual monomers, those are the amino acids, we start bonding them together to make polymers, and you'll often hear the term polypeptide. So polypeptide is coming from poly meaning many, and then peptide is the bond that gets formed when we link these guys together. So when I take one amino acid, go through a dehydration synthesis reaction, remove that water, and link them together, I form these peptide bonds. So that's what you're seeing here in this. So take the OH off of one hydroxyl group, take the hydrogen off the amino group, make the water, and we get a new peptide bond that forms down here. So polypeptide chains really can widely vary in terms of the sequence of amino acids I put in order, as well as how long those amino acid chains become. And if you remember your protein synthesis, this is all driven by your DNA. So your DNA has a set of instructions on what amino acids to put in what order and how long that chain should be. Interesting thing is that these polypeptide chains do have order to them, do have ends to them, so we know where the start and the stop is. And that is because of that amino and carboxyl ends. So when we talk about a free N terminus, all right, that's where your free amino group is. Kind of think of this almost like the capital letter in a sentence. You know, this is where the protein structure starts. We read through it, and then you'll get to our carboxyl end, or your C terminus. Kind of think of like that as the punctuation at the end of the sentence. So there is order to this in the way that they get built, and again, that gets driven back by the order of your DNA nucleotides and that set of instructions as to how to put these guys together. So here's the thing though, when we talk about polypeptide chains, the chain itself, the order of the amino acids, is only the first step. A straight chain of amino acids isn't a functional protein. So in order for a protein to do its job in your cells, that protein then needs to create a three-dimensional shape. It needs to fold, it needs to twist, it needs to coil up on itself in order to actually perform its function. And that folding is going to happen spontaneously in your cells because remember, a lot of those chains are hydrophilic or hydrophobic. The amino acids either have that desire to interact with water or they don't. And so since these are being built in your cytoplasm, which is overarchingly composed of water, these guys, as they get removed from that ribosome during that building process, spontaneously form into their three-dimensional shapes because of that hydrophilic, hydrophobic interactions that are happening. The order in which those amino acids get put together and the ability for them to hydrogen bond or avoid water or like water or additional other bonding that can happen between those side groups, those R chains, drives the way that this ultimately becomes its final shape. And those proteins' final conformational design really drives how it works. And if you think about it, it's a lot like a lock and a key. So go back to the fact that a lot of enzymes are proteins, proteins are enzymes. You've got that sort of lock and key idea where the protein has to be a specific shape to work within whatever its job is, whether it's going to bind some sort of substrate like a traditional enzyme, or it needs to fit into a receptor to make a message happen, chemical message happen, whatever the case might be. In order for it to do its job, it has to fit properly. And so it's really important that those proteins fold correctly or their job gets lost. So keep that in mind. So let's talk about how proteins actually build that three-dimensional shape, because it kind of is this stepwise progression when we start divvying out how this works. So when we look at a protein's what we call its primary structure, this just is that unique set of amino acids. What amino acids am I putting in what order? And remember that these aren't random sequences. This is driven by inherited genetic information. We go through that protein sequence of DNA to messenger RNA out to the ribosomes to put those amino acids together. And that whole sort of central dogma of biology of how we transmit and read genetic information and use it is really what we're talking about here with this primary structure. So if some sort of mutation happens in that DNA, that does then impact this primary structure, which is going to have impacts of the rest of the protein folding process. So primary structure is really important. This is just the sequence of amino acids, which is then going to set up the ability to do the rest of this protein folding we're going to talk about here. So your secondary structure then comes in two shapes. Your alpha helix, which is what you see here. So it's that coiling kind of similar to what you would think of as a double helix in DNA, or these beta pleated sheets. And if you've ever made those sort of paper folded fans as a kid, it kind of looks like that, where you're folding one side and then folding the other, and you get sort of this bending, pleated sheet result. These are all a result of hydrogen bonds that are happening between those repeated units in your polypeptide chain. So this isn't necessarily between R groups, but more about how they cross-link between other components of that. And you can have multiple alpha helices, you can have multiple beta pleated sheets in a particular protein. You may have some mixture of the two. So it really can be a diverse set of secondary structures. You can have an alpha helix, and then maybe there's a straight chain for a little while. And then you can have another alpha helix, and then maybe you have a couple amino acids that are straight, and you've got a beta pleated sheet. So there's a lot of variation in these structures in different amino acids, depending on how long they are, so on and so forth. And these amino acid helices and sheets can be hundreds of amino acids long, or they can only be 10. So there's a lot of diversity here in this secondary structure. But regardless, if the amino acids are able to do this based on the way that the protein is being put together, they will have one or both of these particular structures as part of that secondary component. Tertiary structure, this is a result then directly of the interactions between the R groups. And you can have any number of this tertiary structure being held together by these types of bonds. So we can have those hydrophobic interactions, right, where those side chains don't want to interact with the water, so they bury themselves on the inside of the molecule to try and avoid it. They're relatively weak, but if they can maneuver themselves based on where they are in that chain to get to the inside of a molecule and avoid the water, they'll try to do that. You can get those van der Waals interactions where you get those temporary pushing of electrons, and you get those temporary dipole moments where they can interact. You can get hydrogen bonding between polar side chains. You can get ionic bonding between positively and negatively charged side chains. So some of the acidic and basic ones, you'll see some of that ionic bonding happen. You can get disulfide bridges, which happens between those sulfohydryl groups, those SH groups, where they'll lose those hydrogens, and then the sulfurs will link up and cross-link. And those are actually relatively common and relatively strong. They typically happen specifically between cysteine monomers when they get nice and close together. They'll link together. So when you look at a tertiary structure for a protein, it really kind of looks like a rat's nest if you don't really understand what you're looking at. So if you've ever sort of looked at a bunch of string that's gotten knotted together, that's kind of what this looks like from the outside until you start digging in and looking for that secondary internal structure. So here's that alpha helix, here's some of those beta pleated sheets, right? And then they all kind of cross-fold back onto each other and loop back onto each other because all of this stuff is happening to make this tertiary structure. Now, some proteins, once they get to this tertiary structure, they're done. They're functional. They're going to go do their job. But in certain situations, proteins need a little bit more to actually do their job. So some proteins can stop at the tertiary. They're functional. They're good. Other proteins get to this quaternary structure stage. Not all proteins get here, but when they do, we're talking about now taking two individual, or maybe three individual, or four individual. There's multiple polypeptide chains now coming together. So if I look at something like hemoglobin here, there's actually 4 subunits, 2 alphas and 2 betas. So there's 4 different polypeptide chains that individually went through that primary, secondary, tertiary structure and now have aggregated together to make a true full functioning hemoglobin molecule. Collagen has 3, all right, and they all pretty much make these alpha helices and then they aggregate together there. So they can be identical subunits. This guy's all 4 subunits, but they're all identical. Hemoglobin has 4 subunits, but 2 and 2 are identical. Collagen has 3, but they're all slightly different. And so there's a lot of diversity when we get to the quaternary structure as well. How many subunits do you need? How, are those subunits identical to each other? So on and so forth. So let's just take a look at an example. So remember, going back to all of this, that in order for your protein to fold properly, that means that I need to put the amino acids in the correct order so that they cross-link in the correct places, right? That they link up to each other, they make the right structure. And that goes back to their primary structure. That primary structure, though, is driven by your genetics. So if you have a genetic mutation in your DNA, that's going to result in a change in your messenger RNA, which has the potential to change the amino acid sequence. And depending on what it does to that amino acid sequence, will then potentially have dramatic effects on the way that those proteins fold. It's not always the case. When we get to protein synthesis later this year, we'll talk a little bit more about things like silent mutations or missense mutations, and really where does that mutation happen in your DNA and how does it end up resulting in the effects on that amino acid sequence can have a huge range of effects, from basically not having any effect at all to creating an incredibly dysfunctional protein. And we're going to talk at the dysfunctional end of the extreme here for a second. And this is an example that AP likes to use a lot because it covers a lot of topics. And that's this idea of sickle cell disease. So sickle cell happens when you take a look at your red blood cells. They should be these nice sort of donut-shaped, flexible cells that carry the oxygen around your body. In the case of a sickle cell disease, they end up producing these really rigid, hard, sickled red blood cells. They don't do a spectacular job at carrying oxygen as a result. They also have the tendency to get stuck in really small capillary beds and things that your normal red blood cells would be able to bend and shape and squeeze through these sickled cells because they aren't bendable, and they've got these like hooks on the end, get stuck, they rip capillaries apart, they have a tendency to do a really nasty job to places like your liver. And so people with sickle cell tend to get run down easy, tend to run anemic, meaning they're not carrying oxygen in their blood properly, so on and so forth. So why am I bringing it up here? Well, part of why AP loves sickle cell as a good model to explain how things function is because what ends up happening is that sickle cell disease is a example of a point mutation, meaning we've changed one letter in your DNA, and what that ended up resulting in is a single change to the sixth amino acid in your beta subunit of hemoglobin. And so remember I just said hemoglobin is 4 subunits, 2 alphas and 2 betas. So sickle cell is a result of a single mutation in that beta chain. So this is what the normal molecule should look like so that it can carry that oxygen and give us this bendable shape. That point mutation in your DNA now caused a change in the amino acid at that 6th position. So it should be a glutamate, it's now valine, and that property of those amino acids has changed. So if you went back to that list of physical properties of amino acids, you would see that they're not going to fall in the same category. And as a result, that now messes with the beta subunits folding. So when they fold together, rather than them sort of staying apart and being able to be squishy and bendable and flexible because there's space between them, they end up crystallizing into this fiber. And so the oxygen carrying properties are reduced, but I also now get this much more rigid, fibrous, hard cell as a result. And all it is was one change in the DNA that caused one change in the amino acid sequence that messed with the folding of the protein enough to now cause this as an end result. Not every mutation does that, but this is a nice example of showing that progression of how all of those pieces of protein synthesis to actual function are connected, which is why AP seems to love this as a example for you to know, understand, learn as a way to illustrate all of those steps. So I mentioned at the start of all this that proteins really are the big workhorses in your cells. They are the ones that do virtually every function. And so you should be familiar with some of these, and a lot of these will come back up as we go through the year. So enzymes, we're going to touch on them here in this unit, and then come back to them when we get to cellular metabolism later this year. So this is one that most kids are familiar with because we tend to teach this a little bit more as underclassmen, but they're going to help speed up or catalyze those chemical reactions either by bringing molecules together to build things or helping to break them down as they move. We'll talk a little bit about your immune system this year, so we'll talk about defensive proteins, things like antibodies, storage proteins. We won't hit this too much, but be aware that those proteins can be used for storage of amino acids to build things. So, like the egg whites in the actual chicken eggs, right? And they'll use those to break that albumin down to have amino acids to actually start building the chick in those eggs. Transport proteins, we'll come back and talk a little bit about cell transport this year and how those can be open chains, they can be pumps, they can do all sorts of things to help us get molecules that aren't easily able to pass through the cell membrane because of that hydrophilic interaction that's going on. And so how do we get other things through? Well, the transport proteins are going to help us do that. We will get to some hormones later this year. So we'll talk about the fact that I shouldn't say most, but a lot of your hormones are protein structure. So how do we end up using that? They'll help to coordinate all sorts of systematic activity in your body. Receptor proteins will come up kind of right around the same time as those hormones. We'll talk about neurotransmitters. We'll talk about the fact that they need to have some signal to go into. Even the hormones themselves will have receptor proteins on cells. How do they actually communicate and coordinate that function? Well, they have to have a way to communicate. And since your cells don't have eyes or a mouth, there's no speech involved, there's no ability to see things, so it's all chemical. And so proteins are a lot of those chemical messengers, and then the ones receiving those messages are also proteins. Contractile and motor proteins, we'll bring this up a little bit as we move things through your cells, but a lot of you may be familiar with things like actin and myosin proteins in response to muscle tissue, especially if you've taken a nat and phys before stepping into this course. So how do we contract things, move things around? Actin and myosin are two big proteins that do that, and most people know those associated with their muscles. But there's other motor proteins involved with the movement of cilia and flagella in cells, being able to move organelles around in your cells. All of that is motor protein stuff. And then just general structural protein. So support things like the keratin in your hair, in your nails, other silk fibers from spiders, and all of that, collagen, elastin, all of that is connective proteins to help strengthen your tissues. Of course, some of that does a really nice job. Some of it gets broken down in our bodies over time. We don't do as good of a job building them. So think of things like the collagen in your skin. Got this nice, supple, elastic skin as a bunch of teenagers. But as people get older, that doesn't hold up as well. Your body gets worse at replacing it as you get older. And so you end up with those wrinkles, which is what all of those lovely beauty products are being pushed to try and increase your ability to produce that collagen, to produce those elastin proteins and things like that to sort of decrease those wrinkles as you get older. So one of the big challenges with proteins in your body ties back to homeostasis. So because these proteins do so much work in your body, they're incredibly important. But the folding of that protein is what's driving its ability to do its job. And yet the bonds that are holding that secondary, tertiary, or even that quaternary structure, if they get to that, are relatively weak bonds. So when we look at that sort of rating system of strength of bonds back in chapter 2, covalent and ionic are at the top end, Then you get things like the van der Waals forces, the hydrogen bonds, and things like that that come below that. And if you go back to what I said about the secondary and tertiary structures in proteins, a lot of what's holding them together are things like hydrogen bonds, van der Waals interactions. They're weak bonds. They're not those strong ionic or covalent bonds, which means that they are much more susceptible to falling apart than an ionic or covalent bond would be. So when we get proteins into a situation where the conditions that are surrounding them aren't optimal, they have the tendency to denature. And when they denature, that means that they unravel. They lose that normal conformational shape that they're supposed to have. And if they lose that shape, they become biologically inactive. They can't do their job because their job is based on their shape. So your body does a lot to maintain things like your pH, your salt concentration, your temperature as part of those homeostatic mechanisms. We're evolutionarily designed to do that because we need to keep our proteins from falling apart. When those conditions come back in play, at least in a laboratory setting, we can get those proteins to renature and go back to their conformational shape. Sometimes it happens in your body for things we don't understand, sometimes it doesn't. But it's really important to try and keep your proteins normal because these denatured proteins can't do their job. So just an example, this is why really high fevers can be fatal. You literally melt your brain, and what you're doing is melting those proteins. You're denaturing those proteins, both in your blood and in your brain. So when we get to saying 103 fever, you should be in the hospital. That's because we're worried about that temperature continuing to ratchet up. If you know anything about how fevers work, the point to that fever is to be a positive feedback loop. So you get a fever because you've got some sort of infection. A lot of times it's bacterial. And those bacteria are made out of proteins just like you are. So your body has evolved this fever mechanism as a way to turn up your body's temperature in hopes of denaturing the proteins of that infection agent, whether it's bacterial, fungal, whatever. And if we can ratchet up your temperature and denature the proteins of that pathogen, then we can stop that pathogen from reproducing, which is what it's there to do. And it's the reason you're getting sick. So if we denature the pathogen, we can hopefully clear the infection. Problem is, because it's a positive feedback loop, the continued persistence of that pathogen continually tells your body to turn the temperature up. Well, if we get to a certain point where your body either isn't clearing the infection or sometimes that feedback loop kind of just breaks and you get stuck in that feedback loop and your temperature keeps going up and keeps going up and keeps going up, you get to the point where it starts to become dangerous and we now worry about denaturing your own proteins. Things like the hemoglobin in your blood, we need that to carry oxygen. Proteins in your brain, so on and so forth. And so we have to be very cognizant, medically speaking. A low-grade fever is actually a good thing. But when you get to that high-grade fever, we have to treat it. We have to get that fever down so that it's no longer potentially deadly to you. So most proteins will go through several intermediate states on their way to that stable final configuration. A lot of these can do that just in your cytoplasm and they're fine. But others require some assistance to get them to finish that final steady conformational folding. And that's where these things called chaperones come in. They are in of themselves protein molecules, and they sort of end up producing this kind of like tubular isolated environment. So you can look at, this is what they look like 3D. They actually have a cap, they have a hollow cylinder in the middle. And what will happen in the case of needing one of these is that that polypeptide chain will actually get released from your ribosomes. It will come into this hollow tubed component of the chaperone. Cap gets attached, and inside here will allow the microenvironment to help be more stable, whether it's salt, whether it's hydrophilic hydrophobic interactions, whatever the case might be, it's not being as influenced by the external environment in here. And so they can help with that hydrophilic environment as necessary. And then once the protein's completely folded, other bonding will happen here, that cap will come back off, and off goes that correctly folded protein. It's pretty cool stuff that we're still learning exactly how it works. But interestingly enough, we see some diseases such as Alzheimer's, Parkinson's, if you've ever heard of mad cow disease, that are associated with misfolded proteins. And there's some question as to whether or not these chaperones are playing some sort of role within that disease process, whether they're not working properly, whether they're just absent entirely. We're still kind of trying to flush that out. But it's kind of interesting to see that when that protein folding misses its mark, it doesn't fold correctly, there are some dramatic alternative pathways that happen, disease pathways that happen. And some of them, like the sickle cell, are driven all the way back to your DNA and mistakes there, but others, like mad cow disease, for example, don't seem, as far as we know, to have a genetic component per se, but rather it happens at the protein folding stage that something goes wrong. So how do we actually determine what the folding looks like in the first place? I'm not going to go into the nitty-gritty details because, quite honestly, I'm not a protein folding expert, but it is kind of interesting to see how this all works from a very basic standpoint. So they will use a handful of things to start determining the way that these proteins come together. And one of those techniques is this idea of X-ray crystallography. It's the same thing that we used, I should say, Rosalind Franklin, Watson and Crick, the way they figured out the structure of DNA. And so you'll take an X-ray source, you will bind your protein into a crystal, shoot the X-rays through that. The crystal diffracts all of that X-ray beams in and around the protein itself. They hit a detection plate. The detection plate produces a diffraction pattern, and then you can look at this diffraction pattern and start understanding different shapes that come out of that. How to read it? No idea. But people who are experts in this is what they do. So that's one option, one piece to the puzzle. Others are to do what they call nuclear magnetic resonance spectroscopy, or NMRs. And this is a similar idea. You'll put your protein in solution. You'll put that solution into a tube. It goes into this giant machine. I'm talking a couple of desk sizes. It's huge. It's a big magnetic tube. Kind of looks like a centrifuge, but much bigger. Lots of magnets. They'll shoot a bunch of things through it. And then out pops a spectra. So it's a bunch of lines on a graph, and you've got to know what those lines mean. For those of you guys who take organic chemistry in college, that's probably something that you'll learn how to do, is to read those NMR spectras. This peak at this set of nanometers means that it's a benzene ring. This set of nanometers means that there's a methyl, and you've got to start piecing those puzzles together to figure out what the actual structure is and how that fits. How many benzene rings are there can be indicative of how tall the peak is, so on and so forth. And then there's just the general bioinformatics and computer programs that we'll actually play around with some of later this year when we get to gene expression and looking at how we can start piecing these puzzles together, modeling these protein structures, and coming up with something like this. And how do we know how your RNA polymerase binds to your DNA? It's because we've done all of this sort of research. Somebody has figured that out, and now we can, here at the high school level, start looking at the models that they've built and start understanding pieces of how that comes together and how we can use that moving forward.